In a recent study posted to the medRxiv preprint server, researchers identified recurrent mutations in severe acute respiratory syndrome coronavirus 2. Background: Coronavirus disease 2019 caused by SARS-CoV-2 typically subsides within a few weeks, but in some cases, the virus may remain for an extended period, and the condition is termed long-term COVID-19. Immunodeficient patients with long-term COVID-19 could be a source for novel variations in SARS-CoV-2 critical to its evolution. Previous studies have speculated that the SARS-CoV-2 alpha and omicron variants could have emerged from long-term infections. The present study analyzed recurrent mutations in SARS-CoV-2 and the frequency of recurrence. The researchers built a dataset comprising 168 SARS-CoV-2 genomic sequences from about 28 patients for the analysis. Genome series associated with patients were included in the study following a literature search for case studies. Besides, the COVID-19 Genomics UK dataset was utilized to obtain other genome series. Genome selection criteria were established, which permitted inclusion of genome series if 1. There were 2 genomes present on the public databases, 2. Persistent long-term COVID-19 for 28 days or more, and 3. Sufficient clinical evidence indicating the immunodeficient state in the study subjects. A CIVET report was generated for the genome series to confirm that the genomes resulted from long-term COVID-19. All genomic sequences within a series were stored in a single multi-FASTA file, with a header for patient identification, with identifiers, and the days since the availability of the genome within that series. Mutation calling of the genomes was automated, and the genomes were aligned to an annotated reference sequence of SARS-CoV-2. Observed de novo mutations, but absent on day 0 of the genome series, were investigated by processing the mutation calls. Findings: the authors found the SARS-CoV-2 spike gene with the highest recurrent mutations, with the following 10 amino acid substitutions. The domains with the highest DNM occurrences were the receptor binding domain of the S gene, the N-terminal domain, 5 occurrences, and the signal peptide, 1 occurrence. RBD had the highest AA loci with DNMs, followed by 5 in the NTD and 1 in the SP. The E484K mutation was the most frequent DNM with 8 occurrences. Clustering all DNMs of this locus increased the number of DNM occurrences to 12, indicating its enrichment for DNMs. Recurrent deletions were observed only in the S genes NTD S67 region, S138 region, and S243 region. S gene, that is just 1/8 of the SARS-CoV-2 genome, showed about 34% of all DNM occurrences and 59% recurrent DNMs. Other non-ORF1ab genes had a lower frequency of DNM occurrences. T30I was the only recurrent DNM in the envelope gene with 6 occurrences, and H125Y in the matrix gene was the lone recurrent DNM. Recurrent mutations in the ORF1ab were more, but relatively fewer than in the S gene. 86 out of 195 DNMs were found in ORF1ab, but it had only 6 of the 21 recurrent DNMs. Comparing recurrent DNMs to the United Kingdom Health Security Agency's variant of concern or variant under investigation definition files revealed E484K substitution as the most frequent DNM with 11 appearances. Of the 21 recurrent DNMs observed in the study, 9 defined mutations for a VOC or VUI. Conclusions: the most significant observation was the DNM frequency of the RBD. The RBD, which is just 2% by the length of the entire genome, constituted about 17% of all the observed DNMs, indicating strong mutational selection in the immunocompromised individuals. Moreover, 12 DNMs were observed for the S484 locus, highlighting a strong selective pressure at this site, with the E484K substitution being the most frequent DNM. All recurrent deletions of the SARS-CoV-2 genome were present only in the NTD of the S gene. The S138RDR2, partly constituting the NTD antigenic supersite targeted by most neutralizing antibodies, had 4 de novo occurrences. To summarize, the researchers identified recurrent mutations associated with long-term COVID-19 in immunodeficient subjects. Most of the observed recurrent DNMs are related to immune evasion, enhanced binding affinity to host receptor, or improved virus packaging. Still, these are, in general, less prevalent in a more extensive SARS-CoV-2 population. Based on these, the authors posit that long-term infection might lead to the selection of mutations, which may partly aid intrahost replication and persistence rather than the general SARS-CoV-2 population, in which mutations are strongly selected for interhost transmission. Important notice. medRxiv publishes preliminary scientific reports that are not peer-reviewed and, therefore, should not be regarded as conclusive, guide clinical practice, health-related behavior, or treated as established information.